From the COVID-19 vaccines that changed the world to the future of cancer therapy and even treating rare genetic diseases, mRNA technology is completely rewriting the rules of medicine. But here's the thing, this amazing technology depends on a microscopic delivery service to even work. So today, we're going to take a super high-resolution look inside the tiny little vehicles that are making it all happen. So what are you looking at right here? Well, these are lipid nanoparticles, or LNPs for short. The best way to think of them is as these tiny engineered bubbles of fat, like microscopic delivery drones, designed to protect super fragile molecules like mRNA and carry them safely right into our cells. And believe me, they are absolutely crucial. Without these LNPs, any mRNA we'd inject would get destroyed by our own bodies in seconds, long before it could do any good. They really are the unsung heroes of this whole mRNA revolution. All right, so let's get into it. Here's our game plan for this explainer. First, we're going to talk about the big challenge in actually making these things. Then we'll look at a really clever technique for sorting them out. We'll see exactly why density is the key to everything. And finally, what all this means for the future of medicine. Okay, so let's start with the core issue. When scientists are in the lab making a batch of these LNPs, it's not like baking cookies where everyone comes out the same. The manufacturing process itself creates a lot of variation. I want you to picture a giant fleet of delivery trucks. Some of those trucks are packed to the brim with precious cargo, the mRNA, but a whole bunch of others are just driving around completely empty. And from the outside, they all look pretty much identical, which, as you can imagine, creates a massive problem for quality control. And this quote from the researchers really just hits the nail on the head. If you can't tell the full trucks from the empty ones, how can you possibly guarantee how potent or safe or effective a medicine is going to be? This inconsistency has been a major roadblock. For the longest time, the tools scientists were using they weren't up to the task. Old school methods, things like dynamic light scattering or even super powerful microscopes, could tell you the average size of the particles in your sample, but they couldn't really see what was going on inside. They just couldn't reliably tell a fully loaded, effective LNP from a useless empty one. So how do you solve a problem like that? Well, this is where it gets really cool. Researchers turned to a classic but incredibly powerful technique and gave it a 21st century upgrade. It's called Analytical Ultracentrifugation, or AUC. Now, it sounds super technical, but the idea is actually pretty intuitive. You know how a centrifuge in a hospital lab spins a tube of blood to separate out the red cells in the plasma? Well, AUC does the same thing, but for nanoparticles. Spinning them at mind-boggling speeds, we're talking up to 30,000 RPM. And that force separates them with incredible precision. But here's the kicker. The real breakthrough isn't just spinning things really, really fast. It's the new brain behind the whole operation. The researchers developed a brand new data analysis model they call the custom grid. This is the software that takes all that raw data from the centrifuge and turns it into these beautiful, crystal clear insights. So here's a quick rundown of how it works. First, they prep the LNP samples in these special liquids, sometimes even using heavy water to help with the separation. Then they spin it, and while it's spinning, lasers are tracking every particle's movement with pinpoint accuracy. The heavier, cargo-filled particles sink down, while the lighter, empty ones might not move as much or could even float. Finally, that custom grid software crunches all the numbers and gives you a complete profile of every single thing in that sample. All right, now for the payoff. Let's get to the results, because this is where you can really see the difference this method makes. This is where the magic happens. Okay, so take a look at this graph. This is what you see when you only look at the size of the nanoparticles. The blue curve is the empty ones, and the red and black curves are the ones loaded with mRNA. See how they're all jumbled together? It's a huge mess. Based on size alone, it's pretty much impossible to tell them apart. But now, look at this. This graph shows the exact same samples, but this time they're sorted by density. It's night and day, a perfect separation. The empty LNPs in blue are way over here, and the loaded ones in red and black are way over there. There is zero overlap. And why? Well, it's because mRNA is dense. Packing it inside gives the LNP a totally unique density signature that this method picks up perfectly. So here's the bottom line, the big takeaway. Size is misleading, but density is destiny. It's the density, not the size, that is the definitive tell-all measurement for knowing if an LNP is actually carrying its therapeutic cargo.
And this new method even reveals something totally unexpected. You'd think stuffing cargo into a nanoparticle would make it bigger, right? A little more bloated? Well, the researchers found the exact opposite was true. Adding the mRNA actually made the particles a little bit smaller and more uniform. It seems like the cargo helps organize and compact the whole structure. How cool is that?
And of course, being good scientists, they had to double-check their work. They compared their new custom grid method, which you see here in blue, against another advanced model, shown in red. And as you can plainly see, their new method gave a much more detailed and accurate picture of what was going on, proving it was the superior tool for the job. So, okay, a super precise spinning machine, some fancy software. What does this actually mean for you and me? Let's wrap up by talking about the real-world impact of this incredible science. Well, for starters, this new approach is just wildly efficient. Instead of having to run a bunch of different, less effective tests, drug developers can now run one single AUC experiment and get the whole shebang. They can see the exact cargo load, the precise size, the mass, the purity, all of it in one shot. And that right there is why this matters so much. By giving us such a crystal clear picture of quality, this method helps make absolutely sure that the medicines we all receive are both safe and effective. It's a huge step forward for meeting the strict manufacturing standards that are required for every drug, from vaccines to the cancer therapies of tomorrow. So yeah, this isn't just some neat little lab trick. As the researchers themselves put it, this work establishes AUC as a foundational, cutting-edge tool. It's pretty much set to become the new gold standard for anyone developing these next-generation medicines, whether they're in a university lab or at a huge pharmaceutical company. This breakthrough really just blows the doors wide open for the future. It gives scientists the confidence they need to design even more complex and targeted therapies. And it leaves us with this really exciting question. Now that we can finally check every single one of those little delivery trucks to make sure it's fully loaded, what amazing new medicines are we finally ready to create?